and got excited to hear everything you can talk about.
Yay, thanks. I'm excited to be here with you guys. Like she said, I'm Nicole. I'm a third year mechanical engineering PhD student here. I'm in Eric Seibel's lab. It's the human photonics lab. And the core of my research is the same as the core of this project. It's creating 3D models to help give surgeons more information about the accuracy and completeness of the procedures that they perform, mostly in otolaryngology, also ENT or rhinology, easier words to say, but also expanding into other disciplines. But I threw up a QR code. This leads to like a Google form for you guys to submit questions if you think of them throughout and then don't want to raise your hand at the end. That's what you can do. I also threw it on a couple random slides throughout if you don't catch it right now, but I'll leave it up for a second. Cool. Okay. So the core problem that we're going to tackle deals with chronic rhinosinusitis, which is the disease of what's called the ethmoid air cells, which is what makes up all the space as soon as you enter your nose, like as far back as you can stick your finger, back to like the center of your brain near where your ear hits and up to your skull base, which is the bottom of your brain. And it's essentially made up of this little honeycomb looking structure if you look at it in 3D. But those cells can become inflamed or have tumors or any other number of diseases. And surgeons have to go in and scoop out all of those honeycomb structures. It affects like 1 in 8 adults. Not everybody needs to get surgery, but if you know people that have like a chronic runny nose, it could be that. And after these surgeries, surgeons are trying to scoop out all the honeycomb structures to completion, but they're only working off of a CT scanner and MRI that was taken before the surgery began. And so they don't have clear guidance on where to stop scooping because all the tissue looks the same. But if you go too far, you could hit the brain, you could hit the carotid artery, the optic nerve, the eye, anything like that. And so they're often overly conservative, leaving behind tissue, like I said, 64 to 96% of these procedures, with 1 to 3% suffering injury to those critical structures like the brain, the eye, carotid arteries, et cetera. But because they're overly conservative, 30% of these procedures have to be redone, which is a high number considering how many people this affects. And here's like a more 3D view of what they're seeing. Sorry for the gore. But on the left is the current view with endoscopes, which is like a camera on a stick, which is what surgeons use to guide them through these procedures. The image all the way on the left, I wonder if I can have my mouse in here. This is what your nose looks like before any procedures have been done on it. So again, if you stick your finger in your nose, the first thing that you're going to hit is the middle turbinate, and that's what this is. And then over here is your maxillary sinus, and then up here is your ethmoid cell, so that's what we're scooping out. But right past this is your eye, and right past this wall, it's like just off screen, is your brain. And it's separated by a 0.8 millimeter thick piece of tissue called the lamina propria. Like 0.8 millimeters is tiny. So you have to be really accurate when you're scooping out all of this disease. And they're essentially trying to scoop out all these honeycomb structures to get to this point. And again, eye is right here, brain is right here, and your carotid arteries and your optic nerve are right behind this back wall. But this is the only guidance they're given. So this is a CT scan that was taken before surgery. You can see the honeycomb looking structures here with these really thin walls between them. This is top-down view. This is the front, so these are the eyes. This is the front of your nose. This is your brain. And this is from the side. So scooping everything out. But they don't get any updates to this imaging as they scoop. So again, that's why they're overly conservative. This is some of the technology that larger companies have tried to invent to solve this problem. The most common type of tool is a electromagnetic tracking system that can track the tip of your endoscope or surgical instruments as you go through surgery. And the thought process with this is if you can see where your endoscope is or your surgical instrument is, if the surgeons go through and sort of trace the walls of the inside of the sinus, they can see on the CT scan what tissue they've taken out and what still needs to be remained. And they can see if any of I wonder if I can go back. Any of these air cells are still remaining. So if they were to trace their instrument here, they would know with the space between there and the brain if they still have more tissue to remove. But the problem with these instruments is that this current standard for clinical utility is 2 millimeters of air. And like I said earlier, most of the tissue separating the sinus from the critical structures is less than 1 millimeter thick. And 2 millimeters is just the baseline. It's what these companies are reporting from data that they've collected themselves. And in practice, the surgeons that I've talked to report errors of 1 millimeter or more. So even if you're in the middle of the sinus, it could say you're in the brain or say you're in the brain when you're in the middle of the sinus, which is really dangerous for obvious reasons. Additionally, with electromagnetic tracking, something can happen called registration drift. So the way that you align the CT scan with the actual patient's head is by putting a little sticker on the forehead right here, and you sort of trace it with the probe to make sure it's calibrated correctly. But as surgery continues, things are getting bumped, things are getting moved, the sticker's sliding, there's like sweat, there's blood, there's liquids, there's saline, there's everything else. Sticker moves, patient gets moved, things get unregistered, which can increase the baseline 2 millimeter drift even, or 2 millimeter error even more. Additionally, with electromagnetism, it has interference with any metal. You can imagine a surgical room in a sterile field, there is stainless steel everywhere, which can interfere with it. All of the surgical tools are made of metal. The surgical bed is made of metal. And it can create this like humming vibration in the tract system that introduces even more air. Because of this, it's not super widely adopted. UW Medical Center has it, Seattle Children's Hospital has it, all of these R1 level clinical institutions have it. But if you get outside of big cities like Seattle, New York, LA, and the more well-funded areas, they aren't having access. And even in R1 level institutions, top trauma centers, only 25 to 44% of otolaryngologists, ENT surgeons don't use these devices because of their inaccuracy, even if they have access to them in the first place. And on the patient level, the median cost of getting one of these procedures is $13,000 base. And that's after most insurances. From a clinical mindset, like from the hospital mindset, one of these tools costs between $100,000 and $500,000 just for a single one. And there's often multiple of these procedures happening every day that have overlap between them. Additionally, if you're not from one of these well-funded areas, if you're on Medicaid, Medicare, only 11% of the procedures are able to have access to this. And that was done in a study studying different implementations of clinical devices depending on income level. So there's clearly a gap in accessibility that needs to be addressed, as well as a gap in utility, being the accuracy that we talked about, the 2 millimeters versus 0.8. Usability, surgeons are not used to having to look at a 3D model that shows 3D path, and their ability to use the technology accuracy is affecting that. The speed, anything that introduces 3D modeling or any additional assistance past tracking, it takes too long. So these procedures, they usually try and get them done in under an hour. Creating 3D models can take between 30 minutes to multiple hours plus, if we're looking at research numbers. Hardware, in the OR, they only really have like the size of this table to store all of the additional medical devices, like a couple square feet. And these additional towers are huge, and they alone would take up another area of the operating room. Exposure, the alternative to these higher tech devices is a interoperative CT. So that'd be getting an updated CT halfway through surgery or after they think that they've completed all of their resection that they need to, getting that updated CT and then seeing how much more they have left. Those are expensive. It exposes patients to more radiation than they need to. And so yeah, we're missing something that is highly capable for this specific problem. So to outline the gap, and this is what we started with when I began my PhD, is outlining what the clinical need is. We have a huge team of otolaryngologists at the UW clinic at the Children's Hospital at Harborview, and this is what they said they needed most. They need safe navigation. They don't necessarily care what form it takes as long as they know how to use it. And it's easily interpretable to them compared to what they already know how to do. Something that enables them to ensure complete tissue removal, so getting out all those honeycomb ethmoid air cells. Cost effective for utility, that's both on the patient side, so that $13,000 number potentially bring it down because they get charged for every additional tool that's brought into the OR, and on the hospital side, that $100,000 to $500,000 price mark, and widely available. So this speaks to the hardware because most ORs have their set tools. They're always going to have an endoscope, which is the camera on a stick. But again, only 11% of institutions have these big towers. So we wanted to make something that ensures that all hospitals in America, worldwide, regardless of their income level or availability, would be able to use our technology. Repeats what I said. So we need a design that centers on clinical need, widespread adoption, and technical accuracy. Outlining it even further. So our outline for cost is cheaper than repeated surgeries, again, $13,000 per, plus the cost of potentially intraoperative CT, which is another like $5,000. I'm not sure what it is clinically, but that's what we have to do for our cadavers. No new hardware, just using the existing endoscope. 7mm accuracy, lower than that 0.8 mark that we're trying to do, but at the very least lower than the 2mm mark that's used by existing clinical systems, and no intraoperative drift. So that's the registration sticker. We want something that's not going to use a sticker like that could have the potential of slipping around during procedures. No radiation exposure, that's to be intraoperative CT, so not exposing people to more radiation there. Surgeons want to show surgical progress and completeness, making sure all the cells are scooped out. Faster than intraoperative CT, which is the fastest and most accurate model that they have currently. And that takes between half an hour to 45 minutes for setup, scan, and then takedown. And then they want it to be easy to interpret. They are currently used to only looking at the preoperative CT scan and maybe a camera track. They don't want to learn anything new. Everyone makes this very clear. Doctors are very slow to adopt things they have to learn. So the way that my team decided to approach this is coming at it with 3D reconstruction using just the endoscope. We need to create both photorealistic models, something that's going to widen the field of view of the standard endoscope, which usually has between a 45 to like 120 degree field of view. When you do the 3D modeling, you can expand that field of view further so they can see the whole surgical scene in one view. And then again, 7mm accuracy, that's super important. We decided to replicate intraoperative CT by updating a preoperative CT. So we have the surgeons do their procedure as much as they can do confidently, and then scan with the endoscope in the surgical field. We create a updated 3D reconstruction and then update the preoperative scan to create a simulated intraoperative CT. I'm going to go into that further because I know that was a long sentence, but it essentially just shows the tissue removal or tissue that's been moved, and then can quantify how close they're coming to those critical structures like the brain, the eye, carotid arteries, etc. And yeah, how much they still have to go. And then intuitive, since it's just updating the CT, it's in a format that the surgeons already know how to interpret. They don't have to use anything new. Everybody's happy. Here are some pictures. So this is of a 3D phantom. This is preoperative, and this is after it's been operated on. So you can see, again, this is your middle turbinate, what you feel when you stick your finger in your nose. This is your maxillary sinus, and this is what we're clearing out past here. So we would create a 3D model before surgery begins, and then after it's over. This is the 3D model that we're able to create. So this is actually like tens of thousands of tiny little points. It's quantitatively accurate. I'll talk about the accuracy later, but we map it onto the preoperative CT scan and then can remove any of these cells that have been taken out. So the green outline in here is what this 3D point cloud is representing. This is how we do it. I'm going to run through this really quickly because I could get in the weeds for a whole nother hour on this. And you guys don't need that, but essentially how we do it, remember our goal was to be able to do it solely with it's called a monocular endoscope. It has only one camera on the end. You could do it with your phone. You can do it with any regular like DSLR camera too, but the endoscopes also only have one camera. So we take in that raw video feed and we sample color and density in frames through a process called neural radiance fields. But essentially it looks at each frame at 30 frames per second and picks out the color and intensity at each pixel, and it can map it frame to frame. So it can say this color and this intensity is the same from this frame to the next frame, or it's similar, and then you can track those points and map them into a initial reconstruction. And this is sort of a low resolution version of what we want eventually. You're still able to, so this is a 3D reconstruction right here. You can see it's pretty photorealistic compared to the endoscopic image here, but there's definitely some aliasing, some issues around the edge. But our sort of thing that we go from here is what takes it from medium level accuracy to high level accuracy. And we take the initial low resolution reconstruction and we simulate stereo views. So a stereoscope, I'm not sure if you guys have heard of this, but a monocular endoscope is one camera, a stereoscope is two. And they sort of work the same way your eyes do, and it's how we perceive depth. So we have two eyes, the same as two lenses, and our brain is sort of always doing this internal calculation where your brain knows the difference, the distance between your pupils, and it also knows the focal depth of your eye. And then from then, you're sort of making a reverse triangle in your head, like basic high school geometry, to tell how far anything is away from your pupils by computing that triangle over and over and over again. So if you know anyone that's like blind in one eye, they probably have really poor depth perception. That's why. But we do a similar thing in here where we simulate from one view, simulate having two, and then we're able to make that triangle over and over and over again in that initial scene and get a depth map. So these are two, this is a disparity map, this is a depth map. The darker colors on here are things that are further away, the lighter ones are closer, and then get our point cloud from there. Sorry for more gore. It's almost over, I promise. But this is the simulated left and right post. You can see in these two, it's like a little bit offset between them, even though it's pretty much the same image. We know in our simulation what that distance is between those two cameras. And then we make the triangles out to each of the points and create a reconstruction. So this is our qualitative 3D reconstruction, different from the point cloud. But this is what would widen the field of view for surgeons. We would take a bunch of these two image pairs, sort of stitch them all together, similarly to like how a panorama works on your phone, but in 3D, and you can see all angles you would need to. And then from there, we have, so this is the initial endoscopic image, so just think regular phone picture. This is the 3D model that we got from stereo pairs, and then this is the point cloud. So you can see the colors sort of look the same if you look at it from a specific angle, but it shows the true depth of the scene. And it's sort of hard to like spatially render it here, but you can almost like map out these ridges in your head to the ridges in this scene. So like there's our front wall, and then these are our little ethmoid honeycomb air cells back to here. And then this is the back wall where your carotid artery lives. But from here, we have to do some more computation before it actually becomes useful. So we have these models, we know they're super accurate, but we have to align them to the CT scans similarly to the registration sticker. But the first step in that is taking it from a point cloud, which might have some holes, some dropouts. It has these empty edges here, if there are any low resolution areas, and turning it into something that the CT scan can decide if we're essentially trying to delineate the inside versus the outside of this reconstruction from an open shell. Because if you have a 3D model of how the surgical scene has sort of opened up, you know that any tissue on the CT scan that would be inside of that point cloud is no longer there. So it's sort of like a cookie cutter. So we're making like a cookie cutter shell of the new surgical field that we're trying to chomp out from the initial CT scan. And this is how we do it. We're using a ray-based method. I also won't get into the weeds here, but essentially how it works is we pick a centroid point in the very middle of the point cloud, and we shoot out a bunch of rays from the center and see what it hits. So if we hit the wall, we know that there's no tissue in between there, but if there's little dropout areas, they'll shoot past it, and we can identify those as holes and dropouts. And then we can sort of draw a averaging line between anywhere where dropouts might be and fill it in. So this red area is area that we've decided is inside of the reconstruction, and then anything outside is outside of it. And so we only want to remove or move tissue on the CT scan that's inside of our reconstructed area. This is for aligning that 3D model to the CT scan. Again, could get into the weeds here, but I promise I won't do much. We choose a fiducial point, which is just a 3D location that we know is the same between the 3D model and the CT scan. So like the middle turbinate, the first thing that your finger hits if you stick it to your nose, like we can reliably pick that out on our 3D models and the preoperative CT scan because it's a anatomic location that will never get moved during surgery. So they don't want to ever remove that. They don't want to touch it because it could lead to more issues for the patient. But the very tip of that, we can identify, pick it out on both the CT and the model, and then align them there. And so like here, we have our shared anatomic fiducial. We can do a rigid alignment, and then we do some like gradient descent, fancy math stuff to have the curves line out. We're trying to minimize the distance between all of the points on our 3D point cloud and the internal points on the CT scan. Okay, but this is what we get after that. This is exciting. So this top row, I don't know if you guys remember the CT scans from the very beginning, but it's the same pictures. So we can see these honeycomb structures. This is the preoperative CT scan. Ignore the green and the yellow. But these are the cells that we are trying to remove. And the green and the yellow are the 3D models that we've decided the inside of them is where tissue no longer is. So we aim to delete any area of this preoperative CT scan that lies within those green and yellow models. So these two lines are the simulated post-op CT scan and the actual post-op CT scan. So since we're doing this in an experimental setting, we can get an actual intraoperative CT scan to check our ground truth. This middle model is the preoperative CT scan, but with these areas deleted. So this is what we made with our algorithm. And then the very bottom row is the ground truth and what the scene actually looked like when you get another CT scan after surgery is over. But you can see they ended up looking pretty similar. There's like some little differences here and there. But overall, operating on this, knowing confidently that you still have maybe extra tissue here or wherever, extra ASL here that you still need to resect is a lot better than working off of something completely unupdated. Oh, and I didn't mention, but yeah, when we were coming up with this capability and speed were our main focuses, same thing with the updated displays. So again, going back to our very first slide of what surgeons currently see when they're operating, again, this is the endoscopic view, this is the CT view, and this is what we're proposing they see. So preoperative CT and then the same slide. So instead of operating off of this, they'd be able to operate off of this and clearly identify what cells are still left. And checking back in with our initial need, cost, we know it's cheaper than repeated surgeries in ICT. We'd be able to cut that $13,000 repeated surgeries, hopefully to zero if we're able to predict what tissue is still left accurately. The cost of an endoscope would also be zero because we know that every operating room already has it. So whatever the cost would just be what we set it at because our solution is purely algorithmic. We require no new hardware. Like I said, every clinic in the world that's performing these surgeries already has a monocular endoscope that's of decently high resolution. The accuracy that we're achieving with these 3D models, I don't think I mentioned it here, we're able to get down to 0.3 millimeters of error, which beat our initial goal of the 0.8. That's the thickness of the lamina propria, and also the 2 millimeter, which is what's currently shown by existing companies. And we've validated that with the ground truth CTs that I showed before. No radiation exposure, it's just cameras. We're able to show surgical progress and completeness. It's faster than intraoperative CT. Like I said, it's about half an hour to get a CT scan. Comparable reconstruction models within like academia take between like 10 minutes to 8 hours to run, but most of them I'd say is like 2. Ours runs in less than 20 minutes. I think we're down to 10 maybe in our latest iteration, but it beats ICT, but this is definitely an area we're still working on. And usability, since we're just showing surgeons the CT scans that they've been used to seeing all along, they don't have to learn anything new. So yeah, I also included what we have had to do to get here. I started my PhD in the fall of 2023. We started out this research in urology, doing 3D modeling of the bladder, and it was purely qualitative. We were trying to identify different lesions and different tumors that could exist there. That's also a big problem. And we tried to take the bladder thing pretty far. We competed in Holloman with it my first year, did not do well, didn't win anything. No one was interested. But once we developed the core algorithm, which was the stereo matching, we realized that quantitative accuracy rather than the qualitative 3D models was where the need really lied, not only for sinus surgery, but also for a ton of different surgeries throughout the body, laparoscopic surgeries, keyhole surgeries in pretty much every discipline. And that's when people started getting interested. We had our first meeting with Commotion, which is UW's technology commercialization wing. I'm sure you guys have heard of it by now. Did an IP pitch to them, intellectual property pitch to them in like spring of 2024 and started filing for, I think we have 3 patents now that summer. And that's when we started trying to make it a startup. Because you can't have a product if you aren't able to get it out there. So we started meeting with doctors, started meeting with patients, people who have been faced with multiple repeat surgeries, because the effectiveness of these surgeries also goes down with every repeat surgery you do. Despite the intraoperative CT scans that you get in between them, but whatever. We did ICOR, which is a NSF program to help with customer discovery, helping you meet with these people. We've met with over 100 potential customers at this point, between academia, clinicians, like I mentioned, everybody in between. DDAC is the Drug and Device Advisory Committee. They help budding startups, especially out of UW, with any business, technology, commercialization advising that you could possibly need. They're very helpful. They sort of sit you in a room and grill you for an hour, but you come out of it a lot stronger. And then this spring, I did Holloman Health Innovation with the sinus, and like she had mentioned, we won 2nd place, which is great. And the same week, we found out we got a WRF phase 1 grant, which is $100,000, maybe 120 after the incidentals, to fund this research fully for at least the next year, which was fantastic, especially in the current funding landscape. We filed more patents, and now we're starting with BRAMS, that's the Biomedical Regulatory Affairs program at UW, to help us start FDA filing. And for you guys, I put some resources up here. I'm not sure how many of you guys are actually starting startups, but I had no access or no knowledge of these resources when I started trying to commercialize this, and it was truly impossible to help myself get it. Foster Team Tuesdays is fantastic. Steve White Right are fantastic for finding a team, finding like-minded people, whether you have an idea or are looking to join an existing team. The Nucleate Activator is great too. Nucleate is a nonprofit program that's worldwide at this point, but they match up people with innovation ideas with people that want to help join a team. They put them through a ton of free training with VCs, with lawyers, with regulatory affairs people over the course of 6 months. And then at the end of their program, you have a chance to compete for money. We Lead, Women's Entrepreneurial Leadership Organization, has matching programs as well, similar to SIBA. The Holloman Health Prototype Fund through Dempsey offers money too. It's only a couple $1,000s, but it can really help get everything off the ground. ITHS, WRF, and Life Sciences, Seattle-based things that have helped fund my research and everyone else's, and then Commotion too. And then I have another slide with a bunch more. But this is the last slide, so I'll leave it up. But legal advice that I didn't know existed. UW has an entrepreneurial law clinic that's free for UW students. If you need help with anything, they're there to help. Also, a lot of legal services. I know of some in Seattle. If you want to reach out, I can connect you. But if you pitch your startup to them and they believe in your idea, they'll sometimes give you up to $10,000 in free legal consulting that you'll then pay back later if your idea ends up making it. But if you don't end up making it, they don't ask for the money back, which is so helpful, especially if you're trying to launch out of UW and trying to deal with self-licensing your patent from Commotion. It's really helpful. Pitch competitions, I'm sure you guys know of those already. The Burke Center and Nucleate have their activator and incubator programs. The DDAC, the Drug and Device Advisory Committee, and the Biomedical Regulatory Affairs programs are both great for regulatory advice. As UW students, you get free access to PitchBook and Gartner, which are super helpful for doing commercialization things. It's sort of like Crunchbase, but way better. And then SCORE and the Seattle District Office both give free business advice. But yeah, thank you guys. On that little Google form too, I have my email, and you can follow me on LinkedIn if you have any more questions. But I'm happy to help you guys out however you want. I have been in the weeds in this for so long. I've garnered a ton of knowledge and advice, and I really want to get it back out there to everybody. But yeah, any questions? Or I can